Good afternoon, everyone. Biosynthesis is one of the process in our body system in which protein gets synthesized in our body system. It's one of the process in which protein gets synthesized in our body system. And before protein can be synthesized in our body system, it has to go through different process of mechanism. It has to go through process of different molecular mechanism. And basically, the process of protein synthesis has to go through the process of central dogma. It has to go through the process of central dogma. And what do I mean by central dogma? The central dogma simply describes the flow of information from the DNA to RNA until it forms a functional protein. So in this process of central dogma, let's start from the DNA serving as a template. The DNA is serving as a template. The DNA is serving as a template for the process of protein synthesis in our body system. So the process of protein synthesis takes place in the cytosol, precisely in the ribosome of every functioning organelles of our body system. Ribosome is serving as a benchmark in which protein synthesis is taking its course in our body system. All right. So we're talking about the central dogma hypothesis, whereby information has to flow from the DNA to form a messenger RNA, which now forms a functional protein. Synthesizing messenger RNA from the template DNA is what we refer to as transcriptional process. The transcriptional process, a lot of enzymes that are involved in this process that will bring about the formation of messenger RNA. And from RNA to functional protein is what we refer to as translational process. Translation. So translation is the process of synthesizing protein in our body system. Translation is the process of synthesizing protein from amino acid in our body system. So the protein we want to synthesize must come from amino acid. And the amino acid is derived from the protein diet we take. So the amino acid now is what the body is using in producing the protein. The amino acid is what the body is using in synthesizing the protein. And for the protein to be synthesized, the amino acid needs to be activated by variants of activating enzymes. So these enzymes are the ones that are responsible for the right? So basically, in the process of biosynthesis of protein, or what we call translational process, the information which are genetic, the genetic information that are recorded in the messenger RNA, are stored in the messenger RNA, must be translated into the language of an amino acid in a polypeptide shape, right? So the information, the genetic information that is stored in the messenger RNA must be translated into amino acid in a polypeptide shape, right? And that polypeptide shape makes our protein, depending on the number of the amino acid that will be needed for, the type of the protein that is required by the body to synthesize at that very moment. All right. And before we go into the detail of the translation process, let's briefly talk about, we want to synthesize protein. What are the ingredients that are necessary for the synthesis of proteins? We need some ingredients, we need some important required ingredients to be used, or compounds to be used in synthesizing your protein in your body system. So number one, we talk about the messenger RNA. Messenger RNA is very, very essential because the messenger RNA is serving as a template. The messenger RNA is serving as a template in which the protein synthesis will take its course, because the information that is needed for the protein synthesis is stored in the messenger RNA. It's stored in the messenger RNA. So therefore, we also need the transfer RNA. The transfer RNA is required as ingredients for the ingredients or compounds that are required for the process of synthesizing protein in a body system. So we also have, apart from transfer RNA, we have the 20 amino acids. The 20 amino acids is also very required for the process of synthesizing your protein. We have also the initiation factor. Initiation factors, elongation factors, we have the termination factors, termination factors. What else? This process required energy. So this process required ATP, GTP, and GTP, and also magnesium sulfate, magnesium ion. The magnesium metal will activate the enzyme, right? So we need also ribosomes. Those are the required ingredients for the synthesis of protein in our body system. Another important thing that is required for the synthesis of protein is amino acid transfer RNA synthase enzyme. This enzyme will catalyze the reaction of the amino acid to bind to the transfer RNA, right? So these are the major basic ingredients that are necessary for the process of protein synthesis in our body system. So messenger RNA, the transfer RNA, the 20 amino acid, the initiation factor, elongation factors, the termination factors. We also need ATP, energy. We need GTP, we need magnesium ion, we also need ribosome, and also is that we need amino acid transfer RNA synthesis. Those are the basic ingredients that is necessary for the processes of protein synthesis in our body system. All right. We started earlier on from the course of this discussion that for amino acid to bind to the transfer RNA, it has to be activated. It has to be activated. So the amino acid binding to the transfer RNA, it must be activated. So let's talk about briefly the process of activation of amino acids. So when we have the amino acid binding to transfer RNA, binding to the transfer RNA in the presence of ATP as energy is going to give us amino acid transfer RNA, amino acid transfer RNA plus AMP plus pyrophosphate. Right, in the presence of amino acid transfer RNA synthesis enzyme catalyzing that reaction. Amino acid transfer RNA synthesis. Synthesis enzyme is catalyzing this reaction. And once this is done, so you've gotten your amino acid transfer RNA. The ribosomes, which I said earlier on, serve as a workbench for the process of protein synthesis, has three major sites. It has three major sites. So let's talk about briefly the sites of ribosomes, the sites of the ribosome. So basically, let's also talk briefly about the understanding of the ribosome as one of the organelles we have in all living cells. And we have identified the fact, we have established the fact that the protein synthesis takes place in the cytosol, in the cytoplasm of a cell, in the ribosome, right? In the ribosome. So that is where your protein synthesis do normally take place. And these ribosomes we're talking about, they are made up of two unequal subunits. They are made up of two unequal subunits. So they're made up of large subunits and the small subunits. Large subunits and the small subunits. This is your ribosome. Our ribosome. So this, let's say, for instance, in eukaryotes, we have ATS. In prokaryotes, we have 70 Hz. These are a group of ribosomes, 70 Hz. These are a group of ribosomes that are surrounded with a protein. So for this now, if it has to be broken down for 70S to give you 50S subunits and 30S subunits, right? And this subunit talks about the measurement. See the back, P site. This is the P site. This is the A site. A site. We have three subunits of the ribosome. Three subunits. Three subunit sites, or three sites, precisely. Let's put it that way. Three sites. You have three sites. Three sites of the ribosome. The ribosome has three sites. The E site, which is referred to as the exit site. We have the P site, which is also called the peptidase site. And this site serves as a site for the growing polypeptide chain. Serve as a site, the P site, which is also called peptidase site. The P site is also called the peptidase site. The peptidase site serves as a site for binding the growth of polypeptide chain. The A site is a site where the amino acid transfer RNA binds to. Amino acid transfer RNA binds to. That's a site, right? So those are the sites in which our molecular process of protein synthesis is going to take place. So let's go briefly into the process of protein synthesis. In our body system, or in the molecular knowledge of protein synthesis, the stage in protein synthesis has been divided or categorized into four. So let's talk about the four process or stage of protein synthesis. One, activation. Activation of amino acids. Activation of amino acids. Two, initiation process. Initiation process. Three, we have the elongation process. Elongation process. Four, we have the termination process. termination process. These are the four main stage or process that do occur in biosynthesis of protein. The activation, which we have talked about, whereby the amino acid needs to bind with the transfer RNA to form amino acid transfer RNA in the presence of amino acid transfer RNA synthetase enzyme. So that is activation process. Initiation process. The initiation process talks about the binding of the initiation factor with the ATP to the small subunit of the ribosome, which is a 30S subunit, for instance, in the prokaryotes, right? So when this happens, it activates some cascade of more reaction. And we now have the elongation process. The elongation process is the process of growth of more polypeptide chain. It's the growth of more polypeptide chain. And we now have the termination process. The termination process is a process in which the process of the protein has to stop after it has synthesized its required protein in our body system. If not, there will be abnormal protein synthesis. So the protein synthesized, once it has been, desired protein has been formed, it has to be stopped by way of information, which we refer to as codons of information. Codons of information that are codified in the messenger RNA. Codon of information that are codified in the messenger RNA. All right, so let's start. These are just the major four stage in which the protein process takes place in our body system. We said, one, we have the activation. The amino acid needs to be activated for them to bind to the messenger RNA. It has to be activated. The amino acid needs to be activated for them to bind to the transfer RNA, right? Beg your pardon? So it has to be activated. And now, after the activation, we have initiation process. Then after the initiation process, we have the elongation process. After the elongation process, we have the termination process. Those are the four major stages in which the protein synthesis takes place in our body system. All right, let's quickly go into the proper reaction on how it's being formed. Basically, we said the process of formation of protein or biosynthesis of protein is derived from the information that are stored in the messenger RNA, which needs to be translated into amino acid language in a polypeptide chain format. That's the process of protein synthesis. And in prokaryotes, for instance, that will serve as the foundation for our protein synthesis. The 70S ribosome, is dissociated to form 50S and 30S subunits by initiation factor 3. So dissociation of the 70S to 50S and 30S starts as the initiator process for the formation of protein in a body system. So the 30S subunit, that is 4, which is a ribosome, will go through a process of binding with a ribosomal binding site of the messenger RNA, which is also called Shine-Dagano sequence of the 5' end of the messenger RNA template, right? So this is what we are saying here. Let me depict it here so that you have a better understanding of what I'm actually talking about here. You see, this is your 30S ribosome subunits, the 30S ribosome subunit, which has been dissociated from the 70S subunit in a prokaryote organism, right? A living organism. So the 30S subunit, the 30S subunit requires GTP and some initiation factor 1, initiation factor 2. They bind to the 30S subunit. This is your 30S subunit, which has been dissociated with the help of initiation factor 3. So now the 30S subunit having the GTP binding to it, having the initiation factor 1, initiation factor 2, and also initiation factor 3. Yes, the binding. This is the binding of the initiation factor, the GTP, which is the energy that is required, and also the binding of the initiation factor 3, which has actually performed the function of splitting the 70S into 30S and the 50S. So the small subunit is the precursor in which the protein synthesis actually takes place. So once this is done, this now moves through our messenger RNA. The messenger RNA has a 5' end here, and it also has the 3' end here, right? And along this messenger sequence of nucleotide information, you have what we call the codon, the start codon somewhere there, which is AUG, the start codon, precisely the AUG. And we also have a region here, which is called the Shine-Dagano nucleotide sequence on the messenger RNA. And this is the portion in which the small subunit 30S of the ribosome binds to. That is the end it binds to. So after binding, the transfer RNA, amino acid transfer RNA, which naturally binds to N-formimethionine. It binds naturally, it accepts only N-formimethionine. We bind to region of the codon. You bind to the region of the codon of AUG, and this initiates the process of protein synthesis. And along the line, you now have, along the line of that pathway of synthesis, you now have the ribosome unit with its binding sites. That's 3 binding sites from what we said earlier on. That's the binding site, the E site, the P site, the A site, right? So, and the initiation factor 3 will be displaced out as soon as the messenger RNA is bind. As soon as the messenger RNA binds with the ribosome, the 30S ribosomal subunit. As soon as it binds with it, the IF3, which is the initiation factor 3, is released out as an exit. And by so doing, you now have a ribosomal unit that has the codon, the three sites, right? We have the E site, we have the P site, which is our peptidide site. We now have the A site, which is the amino acid site, which serves as a region for new transfer RNA. Right? Once this is formed, you've been able to make parts of what forms your protein. Then later, subsequently, in this reaction, the larger size or the larger subunit of the ribosome binds to this, binds to the small subunit and the messenger RNA, and that brings about the process of your protein synthesis. All right, so let's talk briefly about, we have been able to establish the process of translation in the context of the fact that it comes from the small subunit. And the small subunit bind with a larger subunit to make this larger subunit complex. All right, so we want to go into the process of initiation. Initiation, the process of initiation. How does it happen? The process of initiation is what we have described along the line with the fact that we have the initiation factors. The initiation factors combined together with the other GTP, for instance, with a small subunit of the ribosomes to form a larger complex, as it were, 30S binding with a 50S to make the 70S complex, the larger protein. So the elongation process now, the elongation process is the process of adding more amino acid to form a longer chain. Adding more amino acid to it, and in the elongation process, we have the binding of the amino acyl transfer RNA. The binding of amino acyl transfer RNA. We also have transpeptidation reaction. And the third one is translocation reaction. These are the three major events that happens in elongation process of protein synthesis. The binding of the amino acyl transfer RNA is the binding of N4-methionine at the P site to the A site. The A site, that's the amino acyl site. Amino acyl site, which requires the N4-methionine, precisely. The transpeptidation is the process of formation of peptide bonds, acyl transfer RNA, and this is catalyzed by transpeptidase enzyme, amino acyl transpeptidase enzyme. Amino acyl transpeptidase enzyme. This is catalyzed by amino acyl transpeptidase enzyme. So therefore, we have the translocation. The translocation is the process of the movement of the ribosome along the messenger RNA until you have a change of the release of the transfer RNA from the A site so that the formed amino acyl transfer RNA can go to the peptidide site. And I said this reaction is catalyzed by amino acyl transpeptidase enzyme. And basically, when this happens, in which the 5' end of the ribosome moves towards when the ribosome subunit moves towards the messenger RNA, getting towards the 3' end, it will reach a region of the stop codon. And once it gets to that stop codon, it's kind of bring about a stop in the reactional process. And we have set it to cap it because I think we've got to the almost end of the protein synthesis, as it were. So to cap it, let's talk about, let's recap on where we start by saying that the process of translation generally starts from activation of the amino acids. Activation of the amino acids, and by way of activation, the amino acid binds to the transfer RNA. And I think one or two things that we did not talk about the transfer RNA is the structure of the transfer RNA. Probably we just need to talk about that briefly. So this is the structure of transfer RNA. It has been said by quite a lot of research that the transfer RNA in structure-wise looks like a clover leaf. So this is the structure of the transfer RNA. And the transfer RNA has 4 functional compounds. The molecule of the transfer RNA has 4 functional compounds. So these are the functional compounds of the transfer RNA. For instance, we have the acceptor sites, which accept the amino acid at the 3-hydroxy prime end. The acceptor site accept the amino acid. Amino acid bind, that's the region in which the amino acid that needs to be activated via binding to the transfer RNA has to bind with the 3-hydroxy end, 3-hydroxy prime end of the transfer RNA. This is the acceptor stem, right? So we also have the D loop. We also have the pseudo-uridine loop. So this is the D loop, this is the pseudo-uridine loop, and this is the anticodon loop. That's the anticodon loop. And basically, the D loop is responsible for binding of the amino acid transfer RNA synthetase enzyme, right? And the pseudo-uridine loop, the D loop is also called dihydrouridine loop. The pseudo-uridine loop binds to the ribosome during protein synthesis. The anticodon loop is a region that binds to the complementary codon on the messenger RNA templates. That's the anticodon loop, right? And this binding is what gives the T transfer the ability to recognize the codon that are inside the messenger RNA for a specific type of amino acid to be synthesized. I think this is just the whole process of protein synthesis, basically. So this is the process in which the protein synthesis gets synthesized in our body system. We have identified the fact that the protein synthesis takes place in the cytoplasm under the organelle called the ribosomes. And basically, the ribosome is also divided into 2 unequal subunits, the larger 50S, the larger small 30S subunits. And these units are being derived from their rate of sedimentation according to ultracentrifugation process. And basically is that for any process of translation to take place, the amino acid needs to be activated by a group of activating enzymes. They're so numerous, and this activating enzyme will allow the binding of the amino acid to the 3-hydroxy end of the transfer RNA. And by so doing, after binding to it, this will kind of initiate the process in with the initiation factors and the energy, which is in the form of GTP and ATP that is required. It also requires magnesium as ion. And what is the function of the magnesium there? Magnesium ions to activate the enzyme amino acid transfer RNA synthesis enzyme and stabilize the whole process of that synthesis. And basically also is the fact that the process of initiation process has to start by splitting of the 70S unit in the prokaryote, whereby the 30S unit is splitted away from the 50S unit by the initiation factor F3, initiation factor 3. So by so doing, this so-called process go on and on, in which the 30S subunit with its initiation factor and GTP and N-formine methionine that has binded to transfer RNA recognize the AUG, which is the start codon on the messenger RNA template. And by so doing, they bind to that region. So once they bind to that region, the next reaction is for the IF3 to be released. T transfer RNA has binded, carrying methionine transfer RNA, which is the only amino acid is recognized, and for methionine compound. So as a result of this, you now have your protein synthesis. And we also said that the process of elongation is a process of growth of more amino acid to form a lengthy polypeptide shell. And in so doing, by so doing, when the binding occurs, the next reaction is transpeptidation, whereby amino acid added and they form a peptide bond. They form a peptide. Transpeptidation is the process of formation of peptide bond, whereby the N-methionine from the P site is transferred to the A site. And this reaction, we said, is catalyzed by transpeptidase enzyme. So, as a result of this, now you now have shifting of the A transfer RNA, which are not charged, away from the P site, so that the new dipeptide bond that is formed can occupy that place, and so on and so on. More amino acid is being added to form a lengthy chain, and that is the process of elongation. And after that is that the protein has been synthesized and it has been formed. The next thing is to stop the reaction, and which is the termination process. So the process of termination precisely talks about the movement of that subunit, the small subunits and the larger subunit that has binded together through the messenger RNA until they reach a region of sequence in the messenger RNA, which is the stop codon. And this stop codon is what we call UGA, UGA, UAA, and UAG. So those are the stop codons. So if the messenger RNA gets to the region whereby it meets with any of these stop codon, it has to stop the process of protein synthesis. And basically, what's actually bring about the stopping or the stoppage of that process is not because it has the anticodon, unlike the process that it started the initiation process. We have what we call the release factor. The release factors are the ones that are responsible for the process of stopping the protein synthesis via binding to the stop codon, the release factor. So once the release factor binds to the stop codon, they kind of degrade the ester bond between the polypeptide chain form and the messenger RNA, and this split the whole process and split the whole compound that has been formed. And this marks the end. This marks the end of this very protein synthesis. I wish you the best, and when next we come again, we'll be talking about more important topics. So do have a wonderful day. And if there are questions, feel free to supply your question. I'll be willing to answer them. Okay, bye for now.